So let's go ahead and look at this process. So the first thing we want to do is we want to get a DNA sample ready to be analyzed. So let's say we have a cell. From that cell, then we extract DNA. Okay. The DNA then has basically chromosomes or even non-chromosomal DNA. And let's say that we have big giant segments of DNA. Okay. Each of these segments then have certain sequence of nitrogenous bases. So let's say A, C, G, T, or something like that, right? And so those sequences then are often consistent between segments of DNA. So if you can isolate a sequence, and we use primers to do this, you can basically chop out a segment of DNA, and you can chop out that same segment of DNA on each DNA sample. Once we chop out a segment, then what we do is we use something called PCR, or polymerase chain reaction. And PCR basically just takes that, amplifies it, and makes, tons and tons and tons of copies of DNA. So now what we have are tons and tons of copies of this same exact DNA segment. We then basically, all of these are going to be the exact same size. If we get different segments from different organisms, then these sizes will vary because this sequence may be longer in a different organism. But for this organism, since it all came from this one, these are all going to be exactly the same size. So next we'll look at how electrophoresis works and how we then analyze these different sizes of pieces from our PCR. So once we have our samples, then we want to analyze them. And in class you're going to use something called electrophoresis. And electrophoresis begins with a rectangular structure which is called an agarose gel. An agarose gel then has the consistency of jello or gelatin. It's made of agarose, which is slightly different than gelatin, although you can run electrophoresis with gelatin if you wanted to. Then what you do is you basically take that agarose, you melt it, you then pour it into a molding tray, and you make a jello mold. In that mold, then you have these little depressions which are called wells. Okay. And then what you do with the gel is you stick the gel in a buffer solution, and that buffer solution then has a charge, and you can run electricity through the buffer solution, and we're going to go from a negative to a positive on that buffer solution. Now DNA has a slightly negative charge, and so having a slightly negative charge, then it's going to go to the positive electrode. And then what we do is we put our samples of DNA into these wells. Now the distance that DNA is going to move depends upon the size of the DNA segment. So if you think about it, think about a tugboat pulling a tugboat versus a tugboat pulling an aircraft carrier. A tugboat pulling a tugboat then is going to go way faster than a tugboat pulling an aircraft carrier. Okay. And it's actually a logarithmic scale, but in this case we'll kind of look at it anyway as a logarithmic scale, but we'll look at it somewhat linearly. So here we have our DNA. Our DNA all has a slightly negative charge. So a tugboat pulling a tugboat, a smaller segment of DNA then is going to go farther than a larger segment of DNA. Okay. In addition, when you get your DNA samples, organisms that are diploid are going to have two different chromosomes, and so when you get two segments of DNA, those segments then are going to come from one parent, the mother, and one from the other parent, the father. If those segments of DNA are the same size, then you're only going to get one band. So these will both be homozygous, but if those segments of DNA are different sizes, you may actually get two bands, which would be heterozygous. So in this case, here's one allele, and here's a heterozygous individual with both alleles, although that's supposed to be straight right there. Now finally, we don't know the sizes of these DNA bands, and so we need to figure out how big they are. And the way we figure out how big they are is we have things called ladders. And then you put a ladder in the final well, and the ladder then has known sizes of DNA that you can just buy online. And so here, let's say we have 1, 2, 3, 4 segments of DNA in our ladder, although let's actually change that a little bit. We'll make this a logarithmic scale. Let's say we have 1, 2, 3, 4 segments of DNA in our ladder. This then is going to be 4 kb. KB, kilo bases, right? 1,000 bases, so that would be 1,000 base pairs, or 4,000 base pairs. 3 kb, 2 kb, 1 kb. So what we can do then is we look at the ladder, we look at our segment of DNA. If we want to get the sizes of these are about there, so maybe 3.2 kb. Right, this one is right here. I guess this is logarithmic, so it'd be what, probably 1.5 kb or so on there. Okay. Usually you would have a computer program that would actually measure the distance that this moved from this, calculate it out, run it on an equation, and you can be really accurate then on the sizes of those particular bands in here. Now for this lab you'll need to know a couple of terms. One is called a deletion, the other is called an insertion. So let's say here is our original segment of DNA. A deletion just means that somehow we had a mutation where this particular segment was taken out and then these two remain. So deletions then end up with a smaller segment of DNA, right? On gel electrophoresis then, if we had a deletion, that segment of DNA will go farther than the original. So if this is the wild type made by a deletion, it will go farther. Then we can have the opposite. Let's say that we started with this one and then we had an insertion. So we basically inserted in here a segment of DNA, let's say right there. Then it's actually going to be bigger. So insertions get bigger, deletions get smaller. Smaller ones go farther, bigger ones don't go as far. Now what I want to show you then is a Punnett square. So for the Punnett square, I'm going to show you a monohybrid cross. So let's say that we have a gene. We'll be looking at a gene that in the mutant form is white eyes. So let's say here's our wild type condition, and we're looking at the white-eyed gene. Okay. And in this case, white eyes is going to be dominant, we'll say. And let's say that, let's say white eyes, which is the mutant characteristic, is going to be recessive. And so we'll do the gene white eyes, we'll indicate it with a prime up there. Okay. Now we'll say that this is dominant and this is recessive. So now if we're doing a monohybrid cross, we're tracking the alleles in a single gene. This gene then is the white eye gene that we're looking at. And so what we want to do is we want to look at the female and the male. And we want to look at the genotype. So let's say this female is white-eyed. The female is diploid, right? Always remember that they're diploid, so it has to have 2 alleles being diploid for the particular gene. And our male then, let's say, is the mutant. So white-eyed prime for our male. Okay. Now what we want to do is we want to make eggs and sperm, right? So we basically want to go through the process of meiosis. And if we take the female and we're making eggs, right, we go through separation. These two would separate, so we're going to have one egg that is white-eyed, or wild-type eyed, and another egg that is wild-eyed. Male is going to be the same. We can only make one type of egg or sperm. So we make our sperm here. And we make our sperm here. Okay? And so we have our 2 types of sperm. This one's really easy. Whenever you do a Punnett square, all you're doing is getting eggs and sperm together. In this particular case, it's no problem because all our eggs are the same, all our sperm are the same, therefore we don't really have to run a Punnett square. We're just going to get that egg and that sperm together. All you have to do in a Punnett square is put egg types and sperm types. If this was a Punnett square, I would just do that, and I would only have one egg type. We'll put the egg on top and only one sperm on the side, and then we have a wide eye and a heterozygous individual then with one allele for the wild type and one allele for the mutant trait in there. Okay, now let's say we want to do a dihybrid cross. Let's say that we have a wild type. And let's do eyes again. So here's our white eye. Let's say we have a mutant. And let's say that equals WE prime. Let's say that we have another trait called hairy abdomen. We have our wild type equals HA and our mutant HA prime. Okay. And so here's one gene then for white eyes. That's our white eye gene. Here's our hairy abdomen gene. This is our mutant with the prime. This is our wild type. We'll just say that the wild type is dominant to the mutant in all these cases, although we're not going to worry about that for this particular example. Now let's go ahead and look at the genotypes. So we want to look at our female. And we want to look at our male. So always when you do the Punnett squares, always figure out the genotypes of your female and your male first, right? Let's say our female is WE, HA prime, HA. So she's heterozygous for the hairy abdomen gene. She is homozygous wild type for the white eye gene. He, let's say, is the opposite. We'll go WE prime, HA, HA. So he is now heterozygous for the white eye gene, and he is homozygous wild type for the hairy abdomen gene. Now what we want to do is make sperm and eggs. So basically we want to go through the process of meiosis. So think about meiosis. These separate, we have independent assortment crossing over. So basically this one could get together with that one, this one could get together with that one, this one could get together with that one, or this one could get together with that one. So if we make eggs, then this egg right here is WEHA. This egg right here is WEHA prime. This one's going to be the same. WEHA prime is right here. This one's the same. We only have two unique egg types. No reason to write four if they're different. We only want unique egg types. Over here we go meiosis with the male and we get sperm. So that sperm would be WEHA and WE prime HA, and there's our sperm. Okay, all we can make are two sperm types from this. We don't necessarily make four sperm types. Now in a Punnett square, all we do is get eggs and sperm together, right? So this sperm can swim over and get together with that egg, this sperm can get together with that egg, this sperm can get together with that egg, or that sperm can get together with that egg. And that's all we're doing with the Punnett square. So we put our eggs on top. And then we put our sperm along the side. And then we just get our eggs and sperm together, right? So that one and that one would go there. That one gets together, that one would go there. That one gets together, that one would go there. That one gets together, that one. And that's all we're doing with the Punnett squares. We're just looking at eggs and sperm getting together. Okay? And we have an equal probability that any of these get together, and that's why we have this ratio in our Punnett square. Okay, and then you can actually fill those in. when you do it. Okay, so now, where students struggle with this is usually in this step going from here to here. Okay? A lot of times what I see from students is a student will have an egg that is a WE, a WE. So they kind of stick these two together. Okay? Eggs are haploid. So diploid means we have 2 alleles from one gene, 2 alleles from another gene. Okay? Eggs are haploid, therefore we can only have 1 allele from one gene and 1 allele from the other gene. So kind of always be thinking about, if you want to double check if you're doing this right or wrong, haploid versus diploid, right? Eggs and sperm are haploid, they're done through the process of meiosis. Okay. In meiosis then we have to have genes for everything. So we have to have 1 allele for this gene, we have to have 1 allele for this gene, because if we had 2 alleles for this gene, we wouldn't have any gene here and that fly wouldn't live at all, right? So this is a no-no. This is what we're looking for, right? 1 allele from one gene being haploid, 1 allele from the other gene.